Okay, let me start with all the key steps. So we started talking about the genetic technologies. If you remember those, a few genetic technologies. But we spent a lot of time talking about recombinant DNA, and highly likely you get a lot of questions related to recombinant DNAs too. So it's good to just go through the key steps of the recombinant DNA first of all. So recombinant DNA, basically we are recombining, we are recreating the whole DNA itself. It has a few main key steps. One, first we identify what the genes here. So in terms of recombinant DNA, I give you some notes for here. So you gotta give things like, actually I need a ruler to separate them. So strongly recommend you to copy this table too, please. It'll be really useful to use. And recombinant DNA. Let's put that in my table. That'll be this. Yeah, let's go for the first. So in gene identification is basically the process that you identify the target DNA. So if you want to get something, we identify first, and then followed by the DNA extractions, and we extract the DNA or the desired the gene. Let's say small letter gene. Okay, from the donor, and then we cut the DNA using the restriction, the enzyme to cut the DNA. And then put it into vector. So we're using the vector of the plasmid. Insert the gene. You insert the gene into the plasmid vector. So it's mainly as a plasmid. Or time to time we may use the some viral vectors too, but commonly we're using the plasmid a lot of times too. Ligation is that basically we're sealing up. So DNA, in case of that, we're using DNA ligase, which is top of the enzyme we use. And also used for the DNA replication process. Basically seals the recombinant DNA itself too. After we get the some plasmid kind of form too, we put it into the host cell. So reintroduce transformations is introduce to the recombinant. Introduce, let's say, the recombinant DNA into the host cells, and the gene expression is basically the host cell is host cells. You think about bacteria themselves too, they replicate and basically express the inserted genes from this process there too. So I just give you some. Okay, I need to go like this. Here, go like that. Just that. Just that. And process the whole things down to here. The last part is therefore, and then what we do is we are for the selection of that, we are use the markers. Now the marker doesn't mean like a marking like here. We basically using the some sort of the think about antibiotic resistance to see that like whether we are successfully on the copy that what we desired to, and so to test or to identify the successful modifications. So this example. So the meaning of the antibiotic resistance basically means after we modify the whole DNAs of the bacteria, we put it into antibiotics. If the cells, the genes get successfully modified, then we would get the some survival of those specific genes. If that's not really properly modified, then we would probably say that all the cells get died out. Okay, these are the key steps of the simply just whole term of the recombinant DNA. Any questions for this? Try to think simple as possible. Like we identify first, we extract the DNA from that the genes, and then we cut in DNA using the restriction enzyme. Vector we generally use the plasmid. We generally use the plasmid or the viral vectors. Ligase basically cells that recombinant DNA substitute, and we transform them and put it into our testing tube. Any questions for key steps, Hayden? Okay, Aaron, any questions? Now, you probably will be hearing a lot of these related to recombinant DNA, and it is kind of very important to know the difference between those two. They are related, but somehow a little bit different as well too. Finished? Okay, thank you. So let's go for the process or the technology called the CRISPR. CRISPR stands for the CRISPR. So this is a type of the recombinant DNA, but the difference between these two are this is a very specific. So unlike the recombinant DNA, this is identifying. So copy this one down to please. So it identify the very specific DNA sequence. Okay, for the modifications. I may just put some little more space. Modification. Yep. Yeah, there are two more. Right. Huh? Yeah, it's there. Okay, not a problem. Then what do we do? This is kind of different to the DNA extractions too. So for the CRISPR, they extract the target genes using the for the CRISPR editing. It's a type of the CRISPR editing for that too. Now, highlight this fine point, please, too. For DNA, recombinant DNA, we use what? The restriction enzyme. This was the key to cutting the ends DNAs. For the CRISPR process, we're using a very specific enzyme called use CRISPR, CRISPR, the Cas9. It's a type of the enzyme and this is for the precise editing. So if I need to describe what the CRISPR-Cas9 compared to restriction enzyme is, this is like, you may imagine a scissor, like scissor, that we're cutting something. Okay, this would be a scalpel, really sharp scalpel. So that it is actually cutting that what we desire to get, unlike but compared to what we have for the restriction enzymes too. So remember this, CRISPR-Cas9 is one of the enzymes to cutting this DNA for the CRISPR process. And vector is basically using this insertion is we use a directly insert, and then we are directly insert. Cas9 is basically CRISPR to the modified genes. The gene, okay, using the CRISPR. The ligation itself, we use the this is basically we don't use a DNA ligase. In case of that, CRISPR, PR, it modifies the genome with the or without the ligation. Okay, just a bit of that too. Now for the transformation, we also use the CRISPR use the CRISPR PR, okay, to modify the host cell, directly. And after that, the edited gene functioning in edited gene functioning in the modified organisms. And therefore, we are just basically after that, we test for the successful like a gene edit. Okay, so that is basically CRISPR is a type of the DNA recombinant DNA processes too, but you may see like difference between two over here. So let's talk a little more about these two differences. I'll give it a little more of those. So just give me the full name of the CRISPR here. Can you copy this one down, please? CRISPR is known as the clustered, regularly clustered, known as clustered, regularly interspaced, okay, and the short interspaced. So palindromic repeat. It's kind of hard to name this palindromic. The repeat. And special enzyme used for this is known as the CRISPR-Cas19. Cas9 is a very specific enzyme that we use in case of this. Or simply we just say Cas9 itself. So what is basically the main difference between two? So I give you a table between like CRISPR and that. So CRISPR PR to the like let's say traditional, as we said, the recombinant DNA. First of all, in terms of precision, okay, CRISPR is like really high in precisely. We're cutting the very specific, so it basically targets the specific like a sequence for the CRISPR. For those, but the recombinant DNA, it'll be kind of lower compared to the CRISPR itself too. Means that because restriction enzyme would not target a specific site, but it's less control, so restriction enzyme. Okay, that cut at specific like a site, not the sequence, but less control, controls over the insertions. So in terms of the speed of that, in speed of the CRISPR, it is relatively fast. Okay, and it allows the fast gene editing. Okay, with the it actually can use with the living cells. Whereas this would be slower for the traditional recombinant DNA processes themselves too. Okay, and they often requires the cloning itself too. So basically the PCR, the amplifications or the amplification. Okay. Another thing is the flexibility. Hi guys, hello. Flexibility. CRISPR can delete, it can also replace or insert the gene. So it's really the multi-flexibles. Compared to the traditional recombinant DNA, they can only mostly insert only. Okay, the new genes. So this is what I call as the difference between the two in terms of that. So efficiency, would it be efficient to use that? Okay. In terms of the efficiency, this is a high successful. The rate of the combining, the making the create new genes. But in terms of that, most trial error for the recombinant DNA, there'll be most likely most trial and error involved for that too. Guys, copy this one down. I'm comparing all the genetic technologies. So we just finished talking about recombinant DNA key steps. Okay, if you remember what we started last week. So recombinant DNA again means that you go through that we recombine the whole DNA itself by select the genes and we extract the DNA from the cells or the nucleus and then we cut in the DNAs, put it into plasmid mainly too, and then put it in the ligate or paste that with the DNA ligase and transform that into the forms that we want in the host cells. And host cells basically copy and paste in the bacteria forms, and then we get the selection and screening of that too. And I'm just writing down the difference between, recombinant DNA CRISPR for now too. So you will get a lot of questions related to CRISPR. So please copy this one down. In terms of the comparing the recombinant DNA and CRISPR in terms of the key steps and the key difference between recombinant DNA and the CRISPR as well too. It's going to be very useful to understand the difference between two. So in summary, CRISPR is a type of the recombinant DNA. This is more like modernized the DNA cutting gene editing techniques compared to the recombinant DNA traditional methods too, because this is going to be highly efficient and kind of fast speed as we're using the very specific enzyme called the CRISPR or simply Cas9. This is the type of enzyme we use for the CRISPR process. We do not use the Cas9 for the recombinant DNA in the traditional method too. Instead, we're using the restriction enzyme to cut the DNAs too. Okay, copy this. I give it time to copy this one down too. Actually, give it like 3 minutes to copy all the here because you just got here. Take your time. There'll be more insulin production and GMOs coming after. Yeah, give it time to copy this one here. Take your time. cut at specific sites, but less control over insertion. Okay, and yeah. No. Viral, yes. Viral vectors. Okay, so main difference between CRISPR and traditional recombinant DNA. You may say, I may say like CRISPR is more like a modernized gene editing techniques. So it's more precise. It's like a fine technologies compared to the recombinant DNA would be more like traditional methods. It started from 1980s, more like modernized from the 2000s or 1990s. I can't remember exact years though. Yep. Most trial error involved. Okay. You can copy this one down. I leave that on the tables on the board too. Almost done. Almost finished. Getting there. Yeah, this part. Still more. Okay. Hurry up. Hurry up. There are many applications of using the recombinant DNA or CRISPR. I brought two, which is kind of popular you will get a lot of times too. One is about we produce insulin using this recombinant DNA process or technologies or even the CRISPRs too. So it's good to know of all the steps of those. First thing, to produce the insulin by artificially, we identify. Oh, sorry, that should be identify. 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 Identify the human insulin genes. Is the first thing we need to do. So basically, various like researchers will be taking that. Hey, there's specific gene, the section of DNA would take a place of the producing insulin, and what we do is you extract the insulin gene. Now from human DNA. In this time now, after we identify and extract the DNAs, we can either use both. We can either use the restriction DNA, or even you can use the CRISPR to cut the bacterial DNA first of all too, and we do use the bacteria plasmid, and then what we do is insert the insulin gene into into bacterial plasmids. And after that, we use the ligase, which is type of the enzymes, and binds the insulin, the gene into the bacterial plasmid. Okay. After that, we put it into transformation. We insert the recombinant DNA. Oh, let's write it the plasmid specifically. Insert plasmid into. It's good to remember the guys here. We're using the E. coli. So E. coli, it's not the dash, E. coli, the bacteria. We're specifically using that E. coli. Now, E. coli is the kind of bacteria can cause the food poisonings too. So what we do is, we got to do here. So this bacteria is then, as they are produced, they produce the human insulin. But after it produces the amount, good amount of the insulin, what we do is we're using the kill this. So use specifically the antibiotic resistance to resistant markers to select the insulin producing bacteria then. What I mean is some of the E. coli would not contain the insulin. So in terms of we putting the antibacteria or antibiotics, we kill those without the insulins. Only the one with the insulin producing will survive too. This is one of the applications, popular application using the either the recombinant DNA or even the CRISPR. Okay, but when if we use the CRISPR, we use a very specific enzyme called the Cas9. If we just follow to the restriction enzyme, the recombinant DNA process, we're using the restriction enzyme cancel that too. All right, what about the GMOs, the genetically modified organisms too? I mean, there could be many like those. So we talked about BT cotton. It could be like some salmon, some other the features. So first thing we do is we always identify that. So we identify the gene for the desired trait. It could be like, so it's resistant to the insect or insecticiders, or like it could be like some strong resilience. Then what we do is extract again the beneficial genes from the beneficial gene from the organisms. Okay, and then we use the either the restriction, the DNA. Oh, sorry, I must say enzyme, not the DNA, enzyme, or the CRISPR. Okay, to cut in case of the organism DNA, organisms DNA. And what you do is we insert the modified genes into the we actually straight to the crop or even for the animal genomes straight away. And what they do, the ligase Okay, that binds genes into the plant animal genomes, into plant or the animal, the genome. And this goes, so I'm following to the transformation student, and the GMO in then what we do is that we insert the genetically modified the cells into the organisms. So just to show the numbers here, step 1, step 2, 3, 4, 5, I'm up to 6 steps. 7 gene expressions, and the GMO is then GMOs expressed the new the traits. Okay, and then like lastly, the test for the we do the test. We test for those successful like a trait expressions. Trait expression. Those are the final steps. So what are we able to see from the difference between two applications, insulin production, GMOs? Can you see, like, for the insulin production, we use plasmid as a vector? Okay, and we like them to just cultivating and creating more of the insulin from that too, and then we get inserting those produced insulin to the body for this especially patient with the diabetes. GMOs doesn't use the plasmid. It doesn't use a plasmid. In terms of vector or vector insertion there, we insert modified gene into the crops or animal genome straight away. Okay, so after that, they just kind of creating the protein inside themselves too, and the ligase binds with the genomes, and basically this makes them change in the genetic structures. This is I would call the main difference between the insulin production of GMOs too, but both of those can actually use both of the traditional recombinant DNA technologies, or even for the CRISPR as well too. So it doesn't matter which one you choose. I mean, time to time we prefer the CRISPR because it's got to be more precise. It takes less taking less time, highly efficient in terms of the like it's really highly successful rate. But in terms of the what we difference between the insulin and the GMO production is we use plasmids a lot for the insulin production. GMO, we don't use the plasmids. We just directly modify the genes of the plants or the crops or the animals too, and put directly into their genomes, and it just binds up with the ligase, and that's it. That's how we can think about the difference between two here. The reason why I'm giving you 4 different type of those genetic, this kind of genetic like technologies, these are very similar. I mean, they're basically talking about the same thing, which is the main of the recombinant DNA. But the slight little things make you confused a lot of times too. So again, to differentiate the recombinant and CRISPR, this is like a traditional method that we use in the restriction enzyme to cut the targeted cut the DNA using the plasmid or the vector of the viral vectors too. In terms of like the traditional methods too. Whereas CRISPR using the very specific CRISPR-Cas9, which is the type of enzyme, the way I describe that again is this is like working as a scissors. They're cutting the section, like in some sections of genes. This is cutting the sequence of that the DNAs too. So you may think like it's more like a precise, more efficient, more fast too. So as we're cutting the sequence of the DNA, I can insert, I can delete, I can translocate. However, for them cutting that, if you think about cutting with the scissors, you only be like at the section, means that you can only insert. Okay, so I'm telling you the difference between the two, and then basically the same thing. We use a plasmid a lot of times for that too, but both of them can be used to produce out of both of the insulin or even GMOs too. So you will definitely get at least one question related to any of those in here. Okay, there's a chance that you may get questions about the PCR, the amplifications too. Yes, Aiden.
So like all the DNA, you can directly insert it into the person, or is it just
Yeah, you can. I mean, like, it doesn't matter which one you use. GM, production GM is simply using the recombinant technologies, but the difference here is we do not use the plasmid a lot of times. Instead, what we do is we directly cut the organism DNA, and then we insert into modified gene into the crop's genome straight away too. So in terms of we are replacing them, we're inserting them to creating there some sort of the new traits without using the bacterial plasmid. Yes, Eric?
What does it say about restriction enzyme other than D?
Donor.
And what's number 6?
Number 6 is transformation. Okay, so I'm try to give you some different ideas and difference between all those technologies too. I hope you get it. I really do, because we get a lot of questions related to that too. You okay, Aaron? Yep. Yeah, also copy those words down to CRISPR and the traditional like recombinant DNA as well too, please. Can I erase them? Yeah, selection and screening. Yeah. Gene edits. Sorry, I've been squatting for too long, so I couldn't really finish my S. Okay, Hayden, can you like visualize the differences of those four given? Yes.
So, like the insert genetics, like number 6.
Number 6, yeah. And so genetically modified cells into the organisms, yeah.
You already say it's like not taken out there. It's like already like inserted.
Well, no, it's what can you say? There's no plasmid or the bacteria we're using here.
Yeah.
We're taking this we're cutting the desired trait. So that identified gene from the desired trait may not be from the crop. So we cut it, and then we extract the benefit gene from, and then we put it into the some that crop's genome. Okay, and then making that as a cell, or making that as like whole genome itself too. We put those genome into a cell of the nucleus.
Like outside.
Well, it could be in vitro, could be inside. Yeah, and then we just let them just have express the genes. Okay, and then therefore they're getting the traits, their desired traits. Yeah.
Then it says like insert. I thought it was already like inserted before.
Insertion modified cells into organisms.
Yeah.
This is like inserting that into the genes into the genomes.
Yeah. Is that like within like
It's possibly be like that we're taking the genome cell out, modify that, put it back into. Yeah. Yes. Cell delete. Slow often requires the cloning, PCR or the amplifications. PCR again means like if basically we are amplifying the whole collecting samples, maybe not be enough for the many purposes, the main purposes. So we are amplifying the whole genes. Yep.
so the main CRISPR is that
Mm.
CRISPR, since it's precise, you can do insertion and all that. But recombinant DNA, what we do,
Insertions. Yep.
it's like scissors.
Yeah, you may just think of it as scissors. Okay. So think of the difference between like scalpels and scissors again too. Scalpel is really sharp. But guys, when you cut it by the scalpels, by the way, you're cutting this like cells or the connection between cells to cells. Whereas you're cutting yourself with the scissors, it would be like a kind of massive cut. It's a kind of rough cut, you may imagine too. Okay. All right. Let's finish up the chapter 3. I'm gonna talk about some whole organism cloning as well. By the way, guys, this is the process called the gene cloning. That we are only talking about the section of the DNAs get copied, modified, unlike the whole organism itself too. So let's look at your workbook, please. Your workbook page. Page 74, please. Let's talk about some whole organisms, like the cloning itself too. So please remember, like this CRISPR, transgenic, or like a recombinant DNA is all about gene clonings. So what does it mean by the whole organism cloning? It's basically like it's also known as reproductive cloning. Has basically we are copying of copy and paste of the whole organism by themselves too. There are two types of the whole organism clonings. So at the side over here, could you copy this table too, please? I give you some like criteria for this whole organism cloning itself too. So in terms of that, we may have the something known as SCNT. Now, SCNT stands for somatic cell, nuclear transfer is known as the SCNT. We can also have the some whole organism cloning of the artificial the embryo twinning. So what is that difference between two? So somatics are definitions for the SCNT first of all. It is basically the nucleus of the let's say a somatic cells. Somatic cells. Okay, it is transferred into inoculated the empty egg of that too. So important thing is we're inserting somatic cells into an egg. They call the SCNT. Okay, now this is basically like this. Artificial embryo twinning is splitting an early stage of the embryo. So it doesn't really get involved. So basically an embryo splitting is what we can think about artificial embryos twinning of that too. Make a twins of the embryo. Okay, so in terms of process, I'll give you some brief detail of process here. We're going to talk more about SCNT today. Process of that is removed here. So nucleus removed from an egg, first of all. Okay, and then we put the somatic cell. Somatic cell, like a nucleus, is inserted. This makes that we're stimulating the division, and making this too, we implanted into a surrogate, the mother's womb. Sometimes we use a laboratory the wombs too, but this is what we do. Unlike the artificial embryo twinnings, this is using that process of the first of all, the fertilized the egg. It divided into the two identical embryos. Followed by, and both developed What's developed separately and comes after if we implant into or implanted into again the surrogate 2. Okay, so this is the main things, two types of the whole organism cloning. We are copying and paste, not just a specific gene, specific section of DNAs, the whole organism by themselves too. So let's talk a little more about artificial embryo twinning. Next page, please. Kind of describe it well in here. So artificial embryo twinning is from the embryo, we artificially divide that into make it two. Think about making them identical twins. From one embryo, we kind of cut it like artificially too. And then after we get two eggs separated, put it into surrogate mother. But what you'll be able to get is we get only get two, but identical twins can be created. So from one egg, we now get two identical DNA genetic informations. They are twins, but in terms of them making the artificial twins, we can create it. Artificial identical twins. So in terms of that, we're using quite a lot, not a lot though, but we do use them in agriculture cloning a lot of times too, especially if you want to increase the amount of the stock numbers of the cattle. That's something we can do. So livestock breedings, or like even for the biological researches too. Where do we use that? If we think about if we're testing one for one sample and leaving one for the controls too, you will be able to know probably like precise data. Or you may think about this is going to be used to get the more number of the cattle, therefore get the more traits. But limitations. Are we able to control the traits we desire to? It's hard to control the traits that we want to have. For example, that during the process of the cutting the embryo, maybe having these two twins, they may have the undesirable traits that we have in terms of they're not really tall, they're not really getting like in a big time to times too. But good thing is this would have the less ethical issues in terms of like animal rights. I mean, it has some, but it's compared to the SCNT, which we're going to talk about a bit later too, it has less terms of that. But we do not do that for humans though. Guys, think about it. If you are made with this, you may have identical twins artificially made. That'll arise a lot of ethical issues. Unlike the artificial embryo twinning, SCNT, somatic cell nuclear transfer, is actually what we do is that, like it's kind of prohibited. We're not allowed to do it in Australia because this is basically like this. I briefly explain here. I take a cell of yours and Liana. The cell is in like your hair cell. Okay, what we do is we prepare our mother's egg. It doesn't have to be your mother's egg or your egg too. It could be any egg. Inoculated means that we're making that empty in the nucleus. Then what we do is from the cell we collect it randomly. Okay, we take the nucleus out from the somatic cells and then put it into these inoculated like an egg cell. Means that what? The cells doesn't need to get fertilized. Can you see the difference here? This is a fertilized and divided. This does not need to get the process of fertilization because we're taking the cells from nucleus from your somatic cell, which is already being what? The diploid number of the chromosomes in there too. What about the end product? If we are able to put it to the surrogate, we're able to produce the offspring, what about the product? What about the product of that in SCNT? Eric, what do you think? Would it be like a product of the SCNT? If I'm telling you, hey, can I just take your cells and do the SCNT? What do you expect? Yeah, it's not just another organism, it'll be you. Genetically exact same. As there's no fertilization, guys. Remember what I've said here. No fertilization means that there is no variation. You are creating yourself, just yourself. Okay, the whole organism is got to be copied from that too. And this was done in, if I remember correctly, in 1996 using a ship, famously known as Dolly the ship, that we taking the cell out of the one mother, one cells too, put it into surrogate mom, and we actually created the genetically identical the offspring. I shouldn't say offspring because it's not the next generation. We basically cloned the whole organism by themselves too. Okay, and then we're doing a lot of investigation through that. Now, this would have lots of the ethical issue. Think about yourself too. If you're getting copied, means that you get the exact same information about used to. Who to know? Who to know? Like this was real Eric. That could be one ethical issue. Like you have two Erics, who to know who's the real, who's the fake. There's no real or fake from that too, because both are scientifically, genetically the same as well too. So let's answer some of the question for 3.2, please. Let's talk about a bit of like difference between that. First, question 1, please. Let's distinguish between the whole organism cloning and gene cloning too. So for the whole organism cloning, this will be related to that replication of the whole organism or the entire the organism assembles to. Okay, which means from one, if I were one, like some organisms, that two, we are copying a paste. Get two of the identical, the organisms, whether that's the SCNT from one organism. Is how we can think about whole organism like cloning itself too. What about gene cloning? Gene cloning is we're not copying down the whole things by themselves too. This is basically replication of a specific gene, or the DNA sequence. The DNA sequence of the interest. So you may think about from one section of DNA, okay, from the genes, what we do is we replace that with some specific part or interest part here like this too. And this gets copied, the many copies of that too. We get the after modified, so there we get the modifications. Or you may say the recombination. And after that, duplication. Whether that's a GMOs or the insulin productions, this is what we call it as gene cloning process. Okay. Question 2. Let's outline the process of the SCNT specifically. So for SCNT process, I briefly gave you that too, but let's write it down as this here, please. 1, the first process for that is we are collecting somatic cell. The collection. We collect the cell of collection. So it doesn't have to be an egg cell on the gametes too. As long as we get the cell is complete means that you may think of a somatic cell with the diploid chromosomes, or you may think that it has containing the whole information, genetic information, I should say. After we collect the somatic cells, we're getting ready for the egg cell. So egg cell collection as well too. We collect the egg cell and then inoculation of that. Inoculation. So meaning of the inoculation simply means that we are removing, inoculation, not or you, my bad. Inoculations simply means that we are getting the removing the nucleus of the egg cell is what we do. After that, we are basically insertion of the nucleus of somatic cells into the inoculated the egg cell. That's a process that we call too. And then what we do is we don't really put it straight away too. We activate it, activation, and after that we culture that in like some artificial features too. So put it into like a lab, the condition of the same similar to like a laboratory, one mom's room, and then after that transfer. It's not a transfer, sorry, the implementation into the surrogate, the mother. The scary thing is the mother can be the organism that is copied as well too. Means that after giving a birth, it will be say just like you give birth to yourselves too, basically same genetic informations. So that means that it carries a lot of the ethical issues behind that too. And a lot of times this is why it's kind of I must say implementation and the seven, of course, the birth of the birth of the genetically identical individual is what we can expect from that too.
So number 5 is like culture.
Culture. We're culturing as in like cultivating. So put it into like a laboratory and to activate that, we actually put a bit of electric shock, just like to let them awake, and then just let them to go for what the cell division process on that too. Okay, any questions about the whole organism process? Pretty straightforward too. Question 3, last question before we have a break. Why do we do that here? Aaron, why do you think we're doing this kind of risky whole organism cloning? I mean, even it sounds really scary. Like we're copying yourself, copying me, copying everyone in here. Why are we doing that?
You have to like create more hands, as in like be more of yourself in terms of like doing a work somewhere.
But we don't copy humans.
Oh, as an organ.
With organisms.
Okay. Just more like a mass production of like
Yeah.
say food or like
So more like agricultural, can we say? Agricultural, like a benefit, or you may say the advancement that we can get some benefit from out of it too. As you say, we may get the clone. The high quality is made like, especially the high quality. The high quality, like the traits, could be possible too. For example, something that we want to inherit, something we want to get like for the next generations too, but we don't really guarantee that we'll get the same traits or not too. So probably think about the easiest way is we're copying that organism by themselves. Okay, now 2, but we don't really do that for the many purposes of that too. This is actually mainly for the biomedical, the researchers in purpose of that. So that's basically like studying for, especially like studying for the genetic diseases. Okay, and it's possible like a developmental biology or like in physiological processes too. Many of those too. And also related to these medications too, especially for the reproductive medicines. Reproductive medicine could be another one too. So it means that we are it's kind of ethical too. Maybe like it's kind of helping us to overcoming like some. It's a wrong way, I must say, but it could be the overcoming the infertility. Kind of related that too, or anything like any of those reproductive challenges that may have. So a lot of times we are only allowed to do this whole organism cloning for the medical or the research purposes. Besides that, for the human good, like think about here. Oh, I need more hands in my farm, so can I just copy that in another person? It sounds wrong, isn't it? Okay. Hey, we need more hands in my factory, so can I just copy the same person, same people too, who is really good at working in this factory here? That'd be wrong. Okay, now this is why like some of the scientific movies, they're using a lot of those. Actually, there's the things about they're copying the same person, which is a great soldier, created the army of that too. Now, that's kind of fictional too, but it's not real.
It's like many organs thing.
That'd be okay. Would that be a whole organism cloning, or more like a gene cloning? It'd be more gene cloning stuff. So we do actually a lot of gene cloning for like that's related to stem cell technology, gen technology too. We're copying the cells of the genes and then kind of like activating that to get the cultivating the organs that we need. That'd be more like gene cloning sections too. But if you wish to do a whole organism, it means that hey, I need a liver, so let's copy Hayden and get the liver out and kill the person. I mean, you get the liver of yours, but that causes a lot of ethical issues too. Do you get what I mean here? So this is why we don't really do that in Australia in many purposes too. There are very specific regions and under the restriction and the legislation under the government and the national levels too. International level actually goes to. All right. Any questions about this? I mean, recombinant DNA technology, we covered everything last week, basically. So I gave you that details in the beginning of the class. A lot of details. So type of question you get is basically like you need to sometimes describe the process of those. It could be whole organism or SCNT. It could be like recombinant DNA. It could also be like CRISPR. I don't know, because I'm not making your school exam paper. But whichever you comes out, you better get prepared for them too. This is the probably the only, I would say probably the question you will get a lot, not in your trials and but in the HSC too. Any questions about this chapter? I mean, this whole modules now. This is the end of module 6. Yeah, we finished module 6 technically. Has anyone tried the worksheet that I gave you last week? Try to finish kind of doing it, kind of finishing it too. Guys, I strongly recommend you to do those questions. This has got to be really, really, really useful. I know it's a thick, fat worksheet and you need to finish too. Do some readings, do some more time too. I only have like 2.5 hours every week too. So I don't want to talk about recombinant DNA every single week. Do some study and get some memorize some stuff you need to do, and it will basically make your jobs much easier later on. All right, let's take a break. Okay, question 14. We're given some information about GM crops, and that's giving some ideas about BT genes and eggplant, all that too. There are two types of the crops given. One is a GM sorghum, and the next one is a BT eggplant. So let's talk about this here. GM sorghums and BT eggplant, in terms of potential benefits to society, it's kind of given. It's kind of given. So started with from you, Aaron. What's the potential benefit to society for the GM sorghum?
Like increases our food supply.
Hmm?
especially for like places like with more drier and hotter like environments.
Yeah. So how'd you write it? Did you say reducing the strains on the current food supply? But why? By how? By how?
Um, because it is big for like
Hmm.
people. They had to like mass produce it.
Is it really for GM sorghum? What was the purpose of doing that GM sorghum like change?
Use protein for that.
Yeah. And how does affecting those only that? I think there's more. Yeah. Did you write it down? Okay. By reduce strain of the current food supply by increase the high the protein contents and as you said like in the increased digestibility. So that's the result, that's the cause, and we need the justifications for that. What are the justifications? Why? That makes what? That makes the less animal feeds less animal feeds it will be needed. Okay, so that's basically what we call the identification. Identify what the answer is. You get like a reasoning in terms of the reasoning, and it'll be justification, justify after that too. What about BT eggplant? What would be the potential benefit for the society? Hayden, the BT eggplant. Yeah,
Stable health, and I guess you can.
yeah.
It will also increase food supply because it can reason
Wow.
because
Yeah.
it can increase like it generates protein that kills like which is like the bottom.
Really, for the eggplant? Are you sure?
The eggplant produces a protein.
Bacteria. So what's what is the guys? What's the big purpose of we doing this? Like creating BT plant or BT eggplant? Why do we do that? I mean, why do we do that? Kill the pests. So as we don't need to do what? To kill the pests by themselves. Isn't it given? The last thing here have reduced the pesticide increase. It's not only the so it's an increase in yield and reduce the pesticides used by the crop farmers too. So main thing is about this here. It's not just increasing. Yeah, it's decreasing the use of the pesticide by like sorry by like as much as by 92%. Now, how is that? Why is it so important? How will it benefit like in a society? I mean, why is it important like we're not to use pesticides, guys? It's harmful for environment and just the environment. Well, organisms too, like basically like in this the or you may just like the limited use of the pesticide or like pesticide limited as in like a decrease. You may say decreased. I think that's a better word. The decreased use of pesticide would be beneficial for the environment as well as Us, the consumers of the eggplant that we're eating too. It's not just simply talking about, hey, BT plant's good. Yeah, I know it's good, but why? You got to justify clearly to get this one down too. What about social implication? What about social implication that start from GM sorghum? Social implication. What could be a social implication for GM, the sorghum? Can we talk about this, like some ethical issues using the GM genetically modified foods, whether that's just GM or the BT or something too? The hint is in your third paragraph, sorry, the second paragraph and the last sentence too. Can you see that farmers looking to grow the transgenic lines are concerned that they will be required to enter into an agreement with the technology provider to purchase new seed every year? Why could this be a problem? Why could this be a big problem? Yeah. Yeah, as I said, like farming. Okay, will farmer have some ability to create in the GM fruits by of GM crops by themselves? I mean, farmers basically like growing the crops, not the one that doing engineering the crops. Who's doing the engineering part? It's the big tech companies too. So if they say, hey, next year we got the new seed. I mean, guys, this is basically the same as you're buying the new iPhones coming in every year as well too. We got the new tech, we got the new machines too. So you gotta buy it. If you don't buy it, you get a disadvantage of the not buying the things too. So that's what we call the social implication here. So farmers, especially in like developing countries, what do you think? They may not have the financial like it's our support from the government too. They simply don't have money to purchase the new GM fraud and GM seeds every year too. Eric, what would be the consequences of that too? If the farmers cannot buy the GM crops or GM crop seeds?
They like lose out on like yield.
Yeah, lose out the yield. Or otherwise, they may need to use a lot of the pesticides too. So again, that's may increase the yield, but they will have some implication on society as well too. Yeah.
This is like a social implication,
Hmm.
but like the points and the second one was like it said like those terms turning lines and they seem limited use because the ease of pulling this person.
Means that, yeah.
Was
Would that be a societal or would that be societal? Yeah, I mean, in terms of what the biodiversity or in terms of the possible like contamination to the wildlife too, we can talk about that as well. But wasn't that something we actually discussed the last week in terms of the ethical issues and environmental issues using the GM crops? Same question. Aaron, what about BT eggplant? What could be the possible? I mean, it would be similar, but what could be possible social implication using the BT eggplant? Social implication, by the way, doesn't always mean the bad things about it. So, I mean, it could be a good. So, we got to talk about some benefits. Could be relative benefit of that too. So, what could be possible social implications? Aaron, yeah. Did you hear what I said? What would be the social implication of the BT eggplant?
Say it's like it's a bit expensive, like in terms of like finance. It's
Yeah.
like financially draining.
Uh-huh.
Like, they may not always be able to like produce the BT eggplant because like it consumes a lot of like money
Money is in what? When to where?
going to the eggplant to like keep producing or keep like well because it says at the bottom it says like
Mm.
they want to avoid the evolution of insect resistance to BT toxins, so like they have to constantly like make sure like the BT eggplant always has like resistance or like acts as a pesticide.
I would not think the pesticide would do anything with the BT eggplant unless it's causing some chemical mutation inside of that too. But what do you mean by we have to take control of that? Carefully manage to avoid the evolution of insect resistant to, yeah. Is that the farmer's job? Taking care of the BT eggplant?
Yeah, it's mainly farmers, and not just farmers, but like maybe the scientists that are researching as well.
It's more like the engineers or the biomedical engineers that are taking control of that too. Yeah, but that could be one thing. They need to control that. So may get the point, but not straight to that because yes, you gotta take some cost to avoid any of the evolution, the insect resistance. But can you like be a little more specific? What kind of like what kind of the management that to go through as well too?
Just like constant, like they have to like they have to constantly like engineer it.
Engineer it. Yep, that could be a cost. But now on the farmer side for the engineer side too. What about like a labor cost? I mean, like surely they gotta do someone's gonna do it. Yep.
I would have thought management would have been like
Hmm.
limiting use of BT eggplants.
Why?
Since the eggplants themselves produce the protein that kills the insects,
Hmm.
increasing the abundance of them will expose like more insects, so more chance of natural selection. That's what I was
Oh yeah, possibly. I mean, like you may natural selections. I mean, like you are talking about the possible chance that we get a super bug who are not really killed by the BTs. So in terms of natural selection, we can talk about it, but would that be related to social implications? Social was like I'm talking about society.
What if bug people kills the BT eggplants? The shortages.
So there's a possibility. There's a possibility. Did you want to talk something? Say something?
Just make more, then the farmer just make more money,
Yeah. Why?
then have more yield.
More yield and less use of the pesticide too means
Less labor.
less labor. Good. It's actually less labor there. Like yes, the management would take a little more cost to like managing that, not to get involved too. But I think it's here. Practically, how are you gonna do it? Yeah, I mean, like, how are you gonna like manage to not to go for like evolution of the insect resistance? Is that even possible to do it? I mean, can you like control micro controlling all the insects or the BT eggplant? I don't think it's impossible. In terms of natural selection, it would naturally happen as well too. So it is unable to control that. So rather, you talk about those uncertain factors too. I mean, there's something. Yes, there's a possibility, but there's not gonna definite things. Guys, what's the definite things happen to the farmers? They will highly likely earn some more money. Okay, or like they have a more profits. Due to that, boom of those BT kind of plant means that they don't need to use a lot of pesticide, less management cost, and they don't really need to hire the people for they can put the chemicals onto. It's good. That's something we can. I would say it's a quite positive impact on their well-being and society as well too. Okay, and that actually happens for the BT cottons too. People don't really need to go into the field to work as the BT are basically like resistant to the pests. Last part is future direction for the research then. Okay, let's think about what about the future direction. What can we do, Liana? Let's start with the GM going to the sorghum. What can we do? It's not there. It's not in there. You gotta think. What could be like thinking here? How can we improve that even from that given? How can we improve that? How can we improve this whole like the things to be in terms of the better way? Like think about this was the issue. Issue with the social implication was probably like the companies using like just for selling those seeds and stuff like that too. First of all, don't you think we need to develop some of the more technology, more like affordable prices too? Okay, that's kind of related to like the government regulations, more like a policy makers too. But we may just think about the things. But another thing is, it's more about the environmental changes too. We need to prevent the spread of those genes to the wildlife to prevent any like type of the mutations or the contamination of wildlife as well too. So it is strictly like in a held like now. If you are planting the GM crops, make sure we do like in the isolated area. Okay, or might be like we are doing some kind of genetic engineering to change the genetic structures a little bit too, so that they can't really like cross-secting with or like intersecting with the other like plants and stuff. That's a possibility. For the BT eggplant, I mean, guys, this is not only just the one answer. As long as your answer makes sense, as long as your answer is making like, oh, this is gonna be like hitting the point, you will actually can get a mark. It's a matter of how to convince the marker, how to convince the reader. I will give you a mark if it's something come up with something different to my more sample answers to, if as long as it's making sense. Yes. Oh no, it was a raising hand. Okay, Eric, is it? Okay, why you guys playing with the hands? Like, what about the Okay, we may talk about this here. So crop management practices. So maybe like for the as we talk about BT eggplant too. So we got to make like a registration. How going to manage those BT, like how to control the BT toxins as well too. That's possible. Anything that can be related from the kind of limitation you can talk about in here. 15 again. It's all about ethics, by the way. All about the ethics, even. So again, like questions about BT, and we're given Alzheimer's disease. The AD is the leading cause of dementia. So use the theory of the utilization ethics, discuss how the development of the new biotechnologies has led to the ethical issues, refer to the information the passage in the answer to. What are the criteria of the ethics? As you have a look at your workbook, you probably know. Leona, what's the some ethical criteria? That we have to follow here. You have a workbook? Have a look at have a look at the workbook. It's there. Ethical criteria for the or any of the biotechnologies. One is should be the well-being. We have to look for the well-being of the society. You may think of a well-being of, let's say, individuals, society. You may think about the environment too. I may consider about ecosystem, the well-beings of everything too. So if your biotechnologies are harming any of those criteria too, it could be the individual, it could be the ecosystems too. There are things we have to consider about it. Okay, is there anything else? Individual rights. So in terms of right, as in like there could be a copyright, human rights, human or the animal rights. In terms of that, do you remember like we don't really do the whole organism cloning in Australia? That's violating the human and animal rights. We're not really copying the whole organism by themselves too. Due to that, it is illegally to do it because it can cause a lot of problems. Okay, right as in that too. What about this? Fairness and the justice. I give an example of the those big tech company who can like force selling those kind of seeds of the kind of expensive ones too. That could be one. Anything else? Hayden? Privacy. I mean, could be like human copyright, but right here, privacy. Then we talk about collecting the personal data of yourself, your biological information by themselves too. Your information is probably saved in your the banking system with or without your permissions. That could be privacy issue. Now, what could be a possible problem here? Just you can't imagine. You can just like imagine about that too. What if there's a hacker like taking your information about your genetic informations, decided to making the hey, let's copy this Leanna's information. What if that happened? And that actually happened. The breach of the personal information that can be used in many different purposes too, especially the bioinformations. Yep. Distribution. These I would call like the main four themes too, but there are more. But as long as you talk about these over here, now how many marks for this? 7 marks. You really need to practice on the questions like this a lot, writing down the ethical issues for each of the givens too, because you will get at least 1 question next week for the ethical issues. Another thing is, before you go home, question 16. This was the real trial paper question in. It's a real again. It's a legit like question that we had in trial paper, and it's not so different to what we learned today in terms of all that too, bacteria, all that. Discuss the societal, economical, cultural considerations around the use of GM plants with the reference to tomato plants. Another example of GM plants too. Guys, can you just make a sample answer and try to memorize it if you're not really like confident to write the answer in the given time? Okay. And this is the reason why, Diana, I'm saying there's a high chance that you may get the research task on these. Yeah, this is your task. You gotta complete all of these, please, for your homework next week. I will post up there some sample answer for all the questions over here, so you can refer to that. But there's one thing. Unlike the other science subjects, like I'm talking of physics and chemistry, bio, especially for this topic, has various answers too. And the way you express your answer, it matters. So I really think like your English skills, literature skills comes forward to this kind of marks too. If you think like you're not confident or can write the 7-mark answer for these questions, I strongly recommend to write a sample answer. No, guys, you can use ChatGPT if you like to. I don't mind that. I really don't. I mean, 3 years ago we didn't have ChatGPT. I couldn't tell that too. You guys are blessed that you have now that you can ask them to write some essays too. But you should memorize that to get the full mark of these questions too. They're highly likely these kind of questions will not come out from now on too due to that ChatGPT. But if you have one, you shouldn't lose a single mark. Any questions? Okay, homework for the next week. Some past HSC questions for these chapters. Chapter 2, I think chapter 2 and 3. More practice. I've been giving you a lot of the past papers. Finish the rest of those topic test questions too, please. And as I said, I'll put it up the answers on the Google Classroom for this one then. There will be a half-yearly test next week. Half-yearly means get a module 5 and 6 together combined.